Yep. Hi, I'm doing my thesis study on population genetic structure of the seagrass within the Banc in Mauritania, looking at diversity and connectivity. So there's 3 species of seagrass that are found in the Banc We have first Allelic Richness. It's a tropical seagrass species. It's a subtidal species found in also hypersaline regions. Their distribution covers an extensive geographic range in West Atlantic, starting from southern Brazil to North Carolina, as well as the Caribbean and the Gulf of Mexico. They're a dioecious species and their seeds are found at the base of the plants. Second, we have Seagrass nodosa. It's another dioecious species found in North Atlantic from Portugal to Mauritania. It's found also in the Mediterranean basin and Canary Islands archipelago and Madeira. They inhabit subtidal regions down to depths of 30 meters, depending on light penetration. Their seeds are also found on the base of their shoots. And third, we have Zostilanolte. It's a temperate species widely distributed in Europe from South Norway to Senegal, the southern limit. It's a monoecious species, predominantly found in intertidal areas, and seeds are produced in the stems of the plants. So looking at the reproductive modes of seagrass, they're capable of sexual and asexual reproduction. So vegetative propagation through clonal growth allows for establishment and maintenance of meadows, whereas sexual reproduction favors genetic diversity and is possibly being one of the only ways to colonize bare and highly disturbed sediments. Plants can also be monoecious, meaning they have both the male and female flowers on the same plants, or dioecious, where the male and female flowers are on separate plants. So the area of study is the Banc in Mauritania, and it's one of the most important seagrass regions in West Africa. It provides nursery services to one of the richest fisheries in the world and is an important feeding ground for wading birds and green turtles. The cold upwelling from the Atlantic and the iron-rich west winds from the Sahara make it a highly nutrient-rich area. It's also one of the only regions in the world where these 3 seagrasses exist together. So the objectives of this thesis was to assess the reproductive modes of the 3 species of seagrass in the Banc So wanting to look at what's the most dominant reproductive mode contributing for populations to persist over time. We hypothesized that the favorable nutrient conditions contribute to a high investment into sexual reproduction. We also want to explore connectivity between populations to see if the different seagrass meadows are reproductively isolated or are they a single connected population. We hypothesized that the complex word circulation contributes to a more genetically isolated subpopulations in the Banc So this is the sample map of the Banc It was sampled haphazardly. These green dots represent where Seagrass nodosa was sampled, so it's sampled throughout the Banc here. The blue dots is Zostilanolte, so we have in the Argan Islands, Kiji and Magma, and the yellow dots is where Allelic Richness was sampled. So just looking at some of our methodology, the data exploration and analysis, we used microsatellite markers that were previously tested and developed and amplified using PCR. Strand and EMSA allele programs and R packages that was used to explore and clean up the data. Clone and genetics was programs to explore the different population genetic parameters, and we use structure and DAPC, or discriminant analysis of principal components, to infer population genetic structure. So looking at some results. Here we have clonal diversity. So clonal richness, clonal richness is a necessary component of genetic variation for organisms with mixed modes of reproduction because it predicts the balance of effective sexual and asexual reproduction over generations. So here we've got some pie charts that represent the clonal richness between the populations in Seagrass and nodosa. So here in the north, in Nudabu, we have high sexual reproduction, so high clonal richness here. You can see that it starts to decrease a bit, with the lowest being in Mojdakna here, and Bay Saint-Jean and Iwik. So this indicates to us that sexual reproduction is dominating in in this species. Next, we have Allelic. Oops, sorry. Next, we have Allelic in the yellow pie charts. Clonal richness was around the middle mark, but the mean value was still above 0.5, which is our threshold. So it still exhibited dominance in sexual reproduction. And the third seagrass species is Zostilanolte. We have a very high investment into sexual reproduction as seen by these pie charts. So here we're also going to look at another parameter or measure of genetic diversity, which is allelic richness, which is measured by the number of alleles per locus. It shows us a species' long-term potential for adaptability and persistence. So a higher allelic richness shows higher evolvability. So even though in a lot of these species, a lot of these populations, the clonal richness is quite high, it's interesting to see that allelic richness is quite low, with the exception of Kiji and Mangma here in Zostilanolte populations. Inbreeding coefficient, or the FIS, is gives us insight into the mating system of the species. So whether they're reproducing at random or with individuals that are more genetically close to them. So a higher FIS value would indicate that more inbreeding is occurring. It's calculated using values of expected heterozygosity under Hardy-Weinberg equilibrium and observed heterozygosity in a population. So the values with the starting limit is values that are statistically significant. So here in Simodosa and Nodosa, we can see that the values suggest that there's more inbreeding happening, with the exception of a few populations like Modjakna. And values for Mamga and Iwik, and Tayshots, it's very close to zero. So we can see that there's more of an equilibrium. For Allelic, it's all negative values, which is contrasting to Simodosa and Nodosa, and it would suggest that more random sexual reproduction is occurring. However, it wasn't necessarily the case. This was more due to fixed heterozygotes throughout the individuals sampled, something that is important to explore and can understand in clonal organisms. In theory, the genotypes of clonal species have no end to their longevity. So they've been around for many generations through many different unfavorable conditions. And what fixed heterozygotes in these populations illustrates to us is that they have been favored by selection over time. So these results show us that. For Zostilanolte, we have also a lot of inbreeding occurring, which is quite expected as it's a monoecious species, so they don't have the same barriers as Allelic and Simodosa and Nodosa to disperse their pollen or seeds further. They tend to just reproduce with the individuals that are closest to them. So you have a higher chance of inbreeding. So looking at some population connectivity, here we have the DAPC plot above, and underneath is the structure. And just a map to remind you of where these samples were, where these individuals were sampled. So here we can see that there's only one main cluster, which indicates that there's a high connectivity between all the populations of Simodosa that were sampled. In Zostilanolte, there's more of a genetic separation between the populations. What's interesting to see is that in Argan Island, here and in Mamga in the north, these populations were more genetically connected than in Kiji, which is between the two. And this was also shown in the structure plot underneath. For Allelic, we can also see that all the populations are highly connected. So what we can see from this study is that there's a higher investment into sexual reproduction found in the 3 species of seagrasses in the Banc. In Zostilanolte, it's extremely high, which is quite rare to see compared to other studies that have looked into the the reproductive modes of the species. And we hypothesized that the areas that Zostilanolte inhabit are more susceptible to disturbances that could occasionally wipe out whole populations. And as I mentioned earlier, sexual reproduction is one of the only ways a species could recolonize bare sediments or highly disturbed sediments. So this high investment into sexual reproduction could be a result of these disturbances. It could also be because it's a monoecious species, so the success of sexual reproduction is higher, so they invest more in sexual reproduction. For Allelic Richness, the mean value also showed that there's a dominance in sexual reproduction, and for Seagrass and meadows, it was varied with these populations. Here, showing more vegetative or more clonal propagation. So a possible explanation for some of the variability that we can see in these populations is looking at the sediment conditions, which is very important for the success of germination of seeds in seagrass. Light penetration could also be another, with more exposed areas could be subject to higher turbidity, which can block some light penetrating to deeper depths. And also grazers, the grazers of the Banc could disturb the areas a lot. Another thing that we looked at in this study is the mating system. So seeing that although there was high investment into sexual reproduction, that the mating system from the inbreeding coefficient suggested there was a lot of inbreeding going on. So this could be due to the fact that the species Seagrass and Allelic, their seeds are found at the base of their shoots, so it limits the seed dispersal, which could be another reason why they tend to breed with individuals that are more close to them. Looking at connectivity in the Banc we can see that it's highly connected between Allelic Richness and Seagrass and Zostilanolte, with the exception of Zostilanolte being a bit more genetically isolated. So possible reasons to this is that because its seed dispersal is highly limited, the connectivity is solely based on pollen dispersal. So even though we hypothesized that the complex water circulation in the Banc could cause more genetically isolated populations, it could be that it helps with the pollen dispersal. And even though grazers could affect the success of sexual reproduction, could also be the reason why how seeds can be dispersed around.
2 more minutes.
Yeah. So just to finish up, this study helps us to better understand the genetic structure of seagrass in the Banc and this is important for population genetic structure and conservation and restoration efforts. Thanks for watching.